Thank you very much, George, and I'd really like to thank you for your steadfast passion, know-how, and commitment towards our shared societal commitment to making a big difference for humanity worldwide. I think we've had a terrific start to the Lausanne Dialogues, and my sincere thanks to all the organizers. These sessions continue to be a groundbreaking way for us to bring different stakeholders together in our shared mission. And this year with the virtual Lausanne Dialogues, I really want to thank everyone tuning in, especially those of you who have loved ones grappling with dementia. I think your partnership is critical, and please be assured that the research community is 100% with you in this fight. And I know that sometimes progress can seem slow and we still have a long ways to go, but I truly, truly believe that we have a lot of reason to be hopeful because we are making progress in many areas that will allow us to eventually conquer these dementias. Following this panel, you'll hear about the many exciting developments in the therapeutic space, but I think it's fair to say, although we're making a lot of progress in tau biology and a lot of emphasis on non-amyloid, non-tau targets, such as inflammation, synapse integrity, neuronal protection, vascular components, and so on, those therapeutic advances are tremendous. But I think what most of us believe that it's almost a truism that even when we have drugs to the right targets, underlying disease biology, we won't make enough progress unless we have the ability to firstly identify individuals who are early on in the disease process where these treatments will have the greatest benefit, to be able to fully understand patient heterogeneity in terms of their underlying disease biology so we can target the right treatments for the right patients, to be able to understand different patients' rate of progression so that we can design clinical studies appropriately, and finally, to have measures that may tell us if our treatments are having their desired biological effect without having to wait extensive periods of time to measure cognitive changes only. So in that context, I'm really delighted to be able to moderate today's outstanding panel, where we brought together some of the world's leading scientists involved in biomarkers and diagnostics in dementia. As you will hear, these individuals and their companies are making tremendous progress in numerous complementary modalities, which includes genomics, fluid biomarkers, and retinal and brain imaging. What I'd like to now do is ask them to briefly introduce themselves before we get into this panel proper. So Mark, if you don't mind, I'll start with you. If you could briefly introduce yourself.
Sure. I'm Mark Mintun. I wear two hats here. I have a hat at Eli Lilly and Company as head of their neuroscience, both the pain and the neurodegeneration research and development. More pertinent to hear that as I'm president of the subsidiary of Lilly, Avid Radiopharmaceuticals.
Great. Thank you, Mark. Steve, if you don't mind, we'll go to you next.
Great, and thanks for having me here today. I'm Steve Verdooner. I'm CEO of NeuroVision Imaging, and we're developing retinal imaging-based biomarkers for detection of amyloid beta plaque, and we also have a fluid biomarker program as well, where we're working on an ATN assay.
Great, thank you. And Richard, if you don't mind.
Hello, everyone. I'm Richard Pither. I'm the CEO of Cytox. We're developing GenaScore, a polygenic risk assessment for future development of Alzheimer's disease.
Next, let's turn to Rianne.
Hi, everyone. My name is Rianne Esquivel. I'm the Scientific Affairs Manager at Fujirebio US, and I focus on CSF and other novel fluid biomarkers for Alzheimer's disease.
Thank you, Rianne. Tobias?
Yes, hi. My name is Tobias Bittner. I'm the franchise lead for Alzheimer's disease biomarkers at Roche and Genentech.
Thank you, Tobias. And finally, the proverbial last but not least, Joel.
Thanks, Husseini. My name is Joel Braunstein. I'm CEO of C2N Diagnostics. We are a technology spinout from Washington University School of Medicine, and we are developing both blood-based and CSF-based biomarkers for early detection of Alzheimer's disease and other related neurodegenerative disorders.
Thank you, Joel. I think as the audience can tell, we've got a world-class panel here. We're going to be discussing various aspects related to diagnostics and biomarkers. And what I'd like to do is ask each of you some questions about your and your company's exciting work. And following that, we'll have an overall roundtable discussion. Hopefully, we'll also have audience participation. So if you have good questions you'd like to submit to us, let us know, and time permitting, we'll try and do our best to answer those questions as well. So perhaps I could start with Mark Mintun. And Mark, I think your organization has been doing some tremendous work related to PET tracers. So I'd like you to provide us an update in terms of the work you've been doing in PET tracers, and then something that you and I know well, but perhaps the audience will benefit from hearing as well. What do you think some of the challenges are to bringing these to the market and to get society to recognize the importance of such diagnostics?
Sure. Avid Radiopharmaceuticals has focused, no surprise, on the two characteristic pathologies of Alzheimer's disease: imaging the amyloid plaques and imaging the tau tangles. I think of developing these tracers as really sort of three major stages. I realize that's incredible simplification, but first stage where you really understand what is the data that supports their use. How good is that? How good is the tracer? How good is the results to compare to the pathology and to the patient's diagnosis and patient's prognosis? The second phase is one we often pass over, but a major component, which is can you build an infrastructure? These tracers are incredibly short-lived. You have essentially when you go to bed, you have no inventory, and you have to make it all the next morning. Build that infrastructure. And then the final one is, once it's out and once it's available, how do people use it? How does it change management? How do we use it in clinical care? How do we use it in clinical trials for research? We of the two tracers, the amyloid tracer is 10 years ago, 8 years ago, has sort of been approved. It's on the market, so it's through that first stage. It built out, and what we've probably focusing on now is that key question of overcoming the barriers. How is it being used? How is it being interpreted? In fact, we were very proud to participate in the IDEAS study, co-sponsored by the Alzheimer's Association, as well as the Medicare, CMS, and other manufacturers of tracers, where the largest Alzheimer's trial, 18,000 people, and it did an amazing job of having essentially doubled the hypothetical rate for how it's going to change management and diagnosis. But the tau tracer is behind. We just recently completed a package and got approval in the United States, and so we haven't built out that infrastructure of making that available. But maybe even more important, we don't yet know how that's going to be used. These two, is it something that needs to be used together? Is it something that can stand on its own as a diagnostic? And I think that's going to be a very important phase. But we've learned a lot about the obstacles of when we were doing the amyloid tracer. The community and us, I think one of the really key things that we learned is, and you're getting to it already, there's a lot of resistance. And say this in a good way. I'm amazed that the medical community overestimates how good they are at diagnosing Alzheimer's disease and underestimates how good real physicians who are taking care of patients and patients and their families can handle the information. I think they really want this information. They really need this information. And when they get it, it changes behavior, it changes lives. So I think one of our biggest obstacles is overcoming that odd double resistance of thinking we don't quite need it, and second, underestimating how much patients and families can use it.
Right, very well said. And if I could just add a couple of, what I might consider editorial comments. So many people in the audience, may have heard that so far we haven't been successful with the amyloid-based therapeutics. Hopefully that'll change. But I think it's very important to recognize that there's almost no question that amyloid does play a very big role in the pathophysiology of dementia or Alzheimer's disease. So having the ability to look at amyloid, whether it's through some of the fluid measures we'll be discussing, or the imaging measures, or the amyloid, or the retinal measures, continue to be very important. And I fully agree with your comment about the very important value of diagnostics and how we collectively need to work as a society. I think certainly right now, with COVID-19, we recognize that had we done more with the diagnostics, we might be in a better position to do things like opening schools, etc. In conditions like HIV until the the ability to measure viral load, it was really still quite challenging to, get a handle. And, dementia, Alzheimer's disease, etc., might be in a similar way. Maybe what we'll do is we'll now move to Steve. And Steve, I think one of the things your group has been doing is some innovative work in retinal scanning technology. So first, I'd like you to perhaps update the audience about what work you've been doing there, and then we'll get into some follow-up questions.
Sounds good, Husseini. So this is work that the foundational work was done at Cedars-Sinai Medical Center out of the Coronial Lab. And when we started many years ago, it was in proving it out in transgenic mice and human cadaver work where they showed a correlation between brain and retina. So what we do is we have non-invasive devices that directly image amyloid in the retina. So maybe a little bit different than blood where we also play, but we directly image amyloid. We know what the morphology looks like. We've done numerous basic science studies, and then we have 24 devices around the world where we're doing clinical studies to further validate this. And this ranges from things like ROSMAP, where we can tie directly to human cadaver tissue, on through validation studies for FDA, which are currently underway. Were underway, I should say, until COVID. So we're paused right now, as are many, but basically we're directly imaging the retina non-invasively with devices that go through a dilated pupil. Those results are sent up to the cloud, they're processed, and then results are set. And essentially think of it as a screen to PET. So we come up with a score. There's a model that we have that we've proven over and over again that we have a reasonably good predictive value. The other aspect that we see in retina is retinal vasculature. So this gives us some clue with respect to vascular conditions in the eye, perhaps one day generating a vascular index, looking at mixed dementias, vascular dementia. So we add that component as well, which we think, is going to be very important as well. So ultimately a screen to PET, maybe in combination with blood.
Great. I think that's very exciting to hear, and I think you almost anticipated one of the questions I'd like to ask you, and I'd like to ask the other panelists when we get to them, is I think it's an exciting time in terms of, brain imaging, retinal imaging, CSF, blood, polygenic risk scores. So maybe you could talk a little bit about what your group is doing related to fluid assay, and also how you're starting to envision sort of combination approaches perhaps even as a screening funnel.
Right. So we basically a few years ago licensed a platform that uses magnetic nanoparticles for detection. So it's a little bit different than in our fluid biomarker platform. It's a little bit different than other platforms that are more perhaps classical. ELISA, it has no complex optics or fluorophores. So this gives us the ability to have very ultra high sensitivity. I'll hold it up to video. We can have props, I guess, no presentations. So it's a chip-based technology. So on each of these fingers is literally a chip that has 80 sensors on each of these chips. So that allows us to multiplex with very small volume of blood, ultra high sensitivity. We don't have as much matrix interference as other things. So it gives us some distinct advantages for low abundance targets. And also really to your point, it's really to create a biosignature. So while we think it's extremely important to determine amyloid status, so to speak, we also think it's important to look, at disease progression and response to therapy. So one can envision an ATN chip, which is what we're looking on, that's looking at amyloid tau and neurodegeneration, 3 flavors of p-tau, total tau, GFAP, neurofilament light, all on a chip in a very small volume of blood. And so we think that this combination approach and creating a biosignature ultimately serves the industry well to not just, because right now it seems that many are fixated for the right reasons on amyloid positivity for inclusion criteria in therapeutic trials. And I think what I'm hoping, and you'll hear it at the end of the program, is that we can move a little bit beyond that and look at a multiplex, whether it's the type of work that Richard is doing at Cytox from a genetic perspective in combination with protein proteomics and other aspects. But really it's to look at that continuum. It's I think very important that we all pivot and think about a combination biomarker approach and getting FDA and pharmaceutical companies to embrace that type of approach. Some already are and doing a very good job of it. But I think we need to move the FDA and the industry. But that's what we're attempting with this platform. We're making really excellent progress. We've done a lot of optimization and analytical validation. We're running clinical samples. We're getting good results. And, stay tuned for 2021. So we're, cautiously optimistic. So.
Great, thank you for those terrific comments. And I think you correctly alluded to some of the fantastic work Richard's group is doing. I'll just mention a couple of things. First is Steve alluded to the ATN, and I'm sure most people are aware of it, but it's one great example of how the Alzheimer's community has come together with the NIH and various stakeholders to really even work on this sort of what one might even think of as a kind of a staging classification where you have amyloid positivity, tau positivity, and the neurodegeneration. So I think that's really allowing us to refine our understanding of the condition. Speaking of refinement, I think we all know that Alzheimer's disease, like most diseases, is multifactorial. And what we mean by that is a strong genetic influence, and then there's other non-direct genetic influences, which could include the vascular insults. We know about head injuries, other things, etc. But we really need to get a handle on the genetic risk to enable earlier intervention and increase knowledge about disease progression. And Richard's group has been doing some terrific work related to what we call polygenic risk scores. So Richard, if you wouldn't mind, if you could both explain the concept of polygenic risk scores and then talk about your work related to polygenic risk scores in Alzheimer's disease.
Well, thank you, Husseini. Yes, unlike the markers that Stephen was talking about, which we would call biodynamic markers that change during the evolution of the disease course, we're all born with a fixed level of genetic risk for Alzheimer's disease. But it's a very complex picture. There are many, many different loci throughout our genome that contribute a little bit of risk and maybe in some cases actually protective. So what a polygenic risk score does, it scans the whole genome and it adds up all the risk associated and all the protective variants into a single score that we call a polygenic risk score. And this was an approach actually that was first pioneered by the Schizophrenia Consortium quite a few years ago, and that's now been applied more recently to Alzheimer's disease. And of course, because we're talking about a fixed level of risk, that assessment can be made at any age. So potentially, individuals can be identified very early, many, many years before symptoms or even biomarkers become apparent in CSF or plasma, which allows you to then plot out effectively a course of diagnostic monitoring for a particular individual with a view then to bringing them into clinical trials at the right time, or indeed managing their healthcare requirements in a very timely fashion. And of course, whether we get true disease modifying therapies in the next few years or not, what we do know is that taking lifestyle and risk avoidance strategies very early on is a very effective way to reduce or push out the time when symptoms might become apparent. So I think, genetics is really coming of age, both in a clinical setting, but also to help, of course, pharmaceutical and drug developers find the right patients to include in their studies at the right time. So our proposition with GenaScore, which takes into account the polygenic component as well as age and gender, is actually to work very much in collaboration with organizations developing more biodynamic markers, be they imaging, CSF or blood-based markers, to pull together, two of those very important features in the overall stratification of those at risk for developing the disease ultimately.
Yeah, thank you very much for the great comments. And I think it's important that you alluded to lifestyle factors as well. So I think that's one of the very important things. I think we all know that, there are certain lifestyle factors which would help us all, whether it's for cardiovascular health or if it's turning out for cognitive health. But it's difficult, for example, all of us to undertake that. I think what Richard's group is alluding to is if you have a polygenic risk score, the good news is that most of these risk genes are what we call probabilistic, not deterministic. So they all have a little bit of risk, and together they may put you at more risk. So, if early on you may know that you're unfortunately one of the people who is at risk, perhaps you could engage in some of those lifestyle measures, so blood pressure, type 2 diabetes, diet, exercise, cognitive engagement. Those are all things which have been shown to have an impact. And I think while we're not suggesting that those things will, prevent Alzheimer's, we still need therapeutics, it could be very important. Richard, if you don't mind, I'll stay with you. I think those of you who've been with the Lausanne dialogue since the beginning today have heard that, unfortunately, COVID-19 has made, things so challenging for so many people and unfortunately caused such a devastation for so many families. But there have been some, I'm almost hesitant to call it benefits, but new ways of working that society has been forced into. So I was also wondering if you could tell us a little bit about your approach to getting data as it relates to remote assessment, particularly during this time of COVID-19.
Sure. Yes, I think one of the best lifestyle avoidance strategies we can all adopt at the moment, of course, is staying at home. But that adds immense pressure to individuals concerned about dementia and of course the people caring for them. So the more we can offer in terms of remote testing, the better. And we know that remote cognitive assessment tools have been with us for some time now and are being more and more refined almost by the month, which is fantastic. But in addition to that, wouldn't it be great if we could do some biomarker assessment to get some very objective data to sit alongside that cognitive assessment tools? So what we do is provide DNA testing from a mouth swab sample if blood's not readily available. And of course, mouth swab kits are very readily available and can be sent to people in their own homes who can then take the sample in a very simple fashion, send it back through the post to the lab doing the assessment. So there's no need to bring them anywhere near the clinic. But of course, two things you need to be able to do this type of remote assessment are, one, a very robust collection strategy, which via these FDA-approved kits, for example. And the other is a very stable analyte. I mean, the nice thing about DNA is that, you can dig up a woolly mammoth that's been buried in the ground for 4,000 years and still sequence the DNA because it's a very robust analyte and not very susceptible to sample handling challenges that we might see with some of the more dynamic and very low concentration markers in both in CSF and in plasma, of course. So, yeah, I mean, it's really been a huge opportunity, I would say, to extol the virtues of these types of approaches during the COVID pandemic and probably beyond.
Yeah, thank you. I have to admit I wasn't anticipating we'd cover woolly mammoths in this panel, but I think it's a great analogy in terms of the stability of DNA. Next, I'd like to turn to Rianne and the exciting work your group is undertaking. To the best of my knowledge, you just received breakthrough status and are submitting to the FDA at the end of the year for your CSF biomarker for the amyloid ratio. If you wouldn't mind, I know most of the things are a journey, so wondering if you could tell us about this journey, what you've learned, and why it's important.
Yes, thank you. We actually just submitted the data for our CSF amyloid ratio to the FDA mid-November, actually. So we expect to hear some news the first quarter of 2021. We're very excited. And Fujirebio has really been in the Alzheimer's disease biomarker space for over 20 years. We acquired InnoGenetics and those CSF biomarkers that are run on the manual InnoTest ELISA. And since then, we've transitioned those markers over to our automated platform on the LumaPulse. And that's really been critical for the ease and consistency that's really required to run a diagnostic on a large scale. And all four of those biomarkers for amyloid and tau are CE marked as well. So they've been run frequently across Europe. And so our FDA submission for the amyloid ratio is really the culmination of years of research into those four markers. And I think a great example of that is we all know, as Richard just mentioned, that A-beta-42 alone is a notoriously difficult protein to work with in the lab. And the ratio can really normalize some of those pre-analytical issues and help to allow for a single cutoff that could be consistently applied across these laboratories across the US.
Yeah, thank you. I'd like to, you triggered a couple of thoughts I'd like to follow up on. You used the term CE mark, so perhaps you could enlighten the audience what that refers to. And secondly, from my understanding, but I'd also like to know if this is what you found as well, there seems to be a reasonable amount of difference geographically in terms of ability to do CSF measures. And, obviously most of us think of it as, something that could really advance the field, but there are different geographical views on that. So in addition to the CE mark question, we could also talk about how you think collectively we can help educate people about the importance of CSF for certain important learnings.
Sure. So for CE mark, broadly, that just means that it's been approved by the regulatory agencies across Europe, so it can be used for diagnostic purposes there. The FDA, of course, has different requirements. very stringent requirements here. We require, first we require a verification trial of our cutoff, and then following that of clinical validation. So our biomarkers have to go through multiple clinical trials to be able to be approved by the FDA and prove that they're robust and reproducible results. And so we've done that here. We saw, above 90% concordance with PET for both sense and spec in our cutoff verification trial. And then we moved that into a clinical validation trial where we saw similarly high concordance in another around 275 patients. And that's how we are able to move forward with that. As for your other question in regards to education, I think the really important thing is really speaking to neurologists, general practitioners as well, and getting them to understand more about lumbar punctures and the safety associated with those, as well as the complications that are associated with CSF collection and the difficulties of the amyloid proteins. Really just having a consistent protocol across these labs is really critical to being able to have, consistent diagnostic results.
Absolutely. And, I think one of the things we're going to really focus on with our final presenters is the impact of plasma, which in my opinion has been one of the really exciting developments in Alzheimer's in recent years. But just wondering what your plans are for advancing plasma measures.
Definitely. And our plasma is a little bit behind CSF just in terms of the research and development and the performance of the markers in plasma because there's such a low amount of protein in the plasma versus the CSF. So we've really seen a lot of progress in just the last year or two across platforms with plasma. And Fujirebio, of course, has been excited about that as well. So we've been working on those plasma markers for Lumipulse as well. We've been looking at, of course, the A-beta ratio in plasma, as well as p-tau181, and then some of the newer markers like NFL and p-tau217. And so as those develop, we hope to be able to bring those to, the general public for Lumipulse as well. Right now, where I see it, they're not they don't have quite as high of performance in the plasma as you do see in the CSF. So we kind of see it like a funnel to CSF and PET testing where you can narrow those patients down that really need that CSF or PET validation for a final amyloid diagnosis. We see CSF in general as an alternative to PET, and that's how our FDA submission is proposed. So the importance there, of course, is having an economical option for these patients and an available option. If there is a disease-modifying therapy coming out in the future, we can imagine the number of patients that would need PET scans. So CSF can be a viable alternative for these patients. But also, as Mark mentioned, until these markers or PET is reimbursed, they're very expensive for the average patient for PET testing. And so CSF can represent an economical option for those patients as well until there is PET reimbursement. And, Mark said it perfectly that this is really important to these patients and their families. So this is a really important option.
Absolutely. It's great that we can keep coming back to what this is all about, which is patients and their families. Tobias, I'd now like to turn to you. So, as I said, I really do think that some of the work with plasma has been one of the most exciting developments recently. Your group is doing some very elegant work with fully automated plasma assays as screening tests. Wonder if you could tell us more about that, as well as the pre-analytic sample handling, and also start to get into what you think is the overall state of plasma assays.
Yeah, thanks so much. I think that's it's a very exciting question. So Roche, as you know, is a pharmaceutical as well as a diagnostic company, and we're the market leader for in vitro diagnostics. We have 30,000 instruments placed worldwide. And the idea is that whatever markers we have on that, and at the moment it's 120 that work in blood, that we can scale this globally. And so the end game for us, or the vision that we have, is to also have these Alzheimer's disease-related markers really for ATN also on that platform and also be able to scale that globally. I think that's really what we're trying to achieve here. And 10 years ago, we have started with the CSF assays when we had therapeutic programs that weren't happy with the assays that were around back then, high variability, and we were just wondering whether there are better alternatives on the instruments that we can provide. And so we have this Alexis platform, which is a high throughput analyzer. It's a Cobas platform called the assays are called Alexis. And we have now developed assays for A-beta and tau for CSF. And about 3 years ago, when the field was surprised by the fact that we were all of a sudden able to measure A-beta 42 and 40, and then maybe about 2 years ago to be also able to measure p-tau variants in the blood, we also tried our assays out on blood, and we've published on this about 2 years ago together with the Swedish group of Oskar Hansson, that these markers and these assays also seem to be working in blood, which was really exciting. And since then, we have been very much invested in improving these assays, also expanding them from the amyloid and tau biomarkers into GFAP, into NFL, also assays to measure ApoE as a protein. And the idea right now, so they are at a stage where, where we call them robust prototype assays. So we have faith in their performance. They're not in vitro diagnostics yet, but they're good enough to run on prestigious and well-characterized clinical cohorts. And that's what we have embarked on for the last couple of months and also for 1 or 2 years. And so we're looking at that data and trying to define the ideal use cases, first of all. And then the second thing is also we're at the moment looking into defining cut points and also defining combinations of these assays that would give us the highest performance possible, so that these assays might eventually not just be used as pre-screening markers, but then also maybe eventually replace CSF and PET testing, because I think that's what we are all striving for. Now, this might take a few years, and these processes are very long, and you mentioned pre-analytics. So this is something that I think the field has been really struggling with for the CSF markers, specifically for A-beta 42. And we have now, with these robust prototypes that we have in plasma, we have done some pre-analytical handling experiments where we tested the impact of multiple freeze-thaw cycles, time to centrifugation, multiple aliquoting steps. And we've published on this, I think, 1.5 years ago. And I think what we found, surprisingly, was that A-beta 42 behaves much better in the blood compared to CSF. There seems to be just one very critical step, and that is really the time to centrifugation. So if you let your whole blood, the drawn whole blood, stand for more than 2 hours on your bench, then the A-beta levels decrease. Other than that, we haven't found any large differences for freezing, thawing, aliquoting, etc. So I think that's really good and might make these markers really fit for routine use, and I think that's quite exciting. So I hope that the field is not going to struggle with plasma markers as much as with CSF markers in the past.
Great, thank you. And I think you touched on a very important point. Again, some people in the audience may not sometimes be aware of just how long these processes are and just how much quality control we need, so that once we've really standardized things and we feel confident, it doesn't matter whether you're in a remote rural area or in a academic center, you can feel confident about the performance. So thank you for that knowledge as well. Now I'd finally like to turn to Joel, and I think it's a very exciting time for you and your company. Wondering if you could share the data, the latest validation for your platform and your vision for post-FDA approval.
Sure, thanks, Husseini. It's great to be here, and appreciate the conference organizers for inviting us. This is an exciting time for our company. We've actually been around since 2008, and so we kind of feel like we're just getting off the ground. It just, I think, underscores the time, attention, resources, commitment, focus requirements to be able to optimize development of assays, biomarkers that we think can have a really transformative impact in the field. And as some of the people attending here may know, some of the underlying technology originated from the Washington University School of Medicine in the laboratories of Dave Holtzman and Randy Bateman. And, the data look terrific in the academic setting, but the challenge is, how do you translate that into a marker that can be industrialized, that can be used across multiple different clinic sites, recognizing some of the variability that may exist in how samples are collected, how they're subsequently sent to a facility for evaluation using the instrumentation. Our test is the test that we just launched, and we believe it's the first blood test to reach the commercial market in the world for use in the clinical setting. And we pursued a CLIA regulatory route, which, as people may know, references a certain laboratory certification that has undergone the appropriate analytical validation requirements for development of the assay. That has all been performed at C2N. And then it's one thing to have a really well analytically validated assay, but you also have to demonstrate the clinical validity. And so that work is the product of about 4 or 5 years of clinical studies. Some of the original clinical validity work originated from the Bittner laboratory, with some publications that demonstrated the high performance of using a blood-based test to measure amyloid 4240 plus apolipoprotein E to predict risk. And then at C2N, we've embarked on, at this point, 3 totally independent studies that effectively demonstrate the reproducibility and robustness of the technique, totaling patients over 1,000. And so the clinical data set that we felt very confident in launching this product into the clinic was based upon 2 studies. One, which Mark already alluded to, the IDEAS study. IDEAS was this nationally represented study that was supported by CMS, the Alzheimer's Association, Eli Lilly, a number of other sponsors that committed to providing their tracers. And what C2N did was we actually performed a sub-study from patients in IDEAS, and using the blood technique, we analyzed those samples, and we were able to correlate the blood measurements with the amyloid readouts that were evaluated under IDEAS. Now, we actually added another component to the IDEAS study, which a lot of the PET amyloid interpretations were based on local interpretation. That's the best way to understand the real-world effectiveness of the tracers. So what we did was we centralized that process, and we had a standardized amyloid interpretation by a vendor that is very experienced in centralized reviews. This was very important for 2 things. One was to try to optimize the quality of your reference standard. So when we have a blood test, we're looking to compare to a reference standard, and in this case, it's amyloid PET. And the other thing is, so that's important to really understand the performance of your test. But also the FDA has these expectations because, again, when you make your claims on the label, what are you proposing to claim? And when you have a good outcome measurement, you have good evidence if the data supports it to make your claim. And so that's kind of where we are. We evaluated samples from what we called the Paris study, the sub-study of IDEAS. And then we actually paired it up with another study of approximately 500 individuals, totally separate cohort, with individuals that had early MCI and mild cognitive impairment, which was similar to the population of individuals who presented under the IDEAS study. And we independently evaluated the performance of the test. And after understanding kind of optimal cut points for determining whether the test is positive or negative or indicative of probable amyloid pathology or not, we were able to apply those cut points to this second independent data set. And we were absolutely thrilled by the very tight correlations between those data. In fact, we saw virtually no drifting of the measurement on the establishment of the cut point. We've actually subsequent done a number of other studies. There's a lot of collaborations in the works. We're in the process of putting together several publications that will be forthcoming shortly that demonstrate some of this validation data. And we introduced the test a few weeks ago, and it's early, but what we've observed is a tremendous outpouring of interest from both physicians and patients. And I think this is alluded to by Mark and their experience with Avid, in that patients want access to information. I think the idea of saying, well, having a dementia diagnosis and maybe there's some fear or concern to get more granular and understand what the condition is, I think is a bit of a historic way of thinking. I think we have to be much more progressive and say patients want to be informed. When patients are informed about their healthcare, they are more proactive. They're more proactive for their family, their planning, their exercise, their lifestyle modifications, and importantly, they understand their potential opportunity to participate in important clinical studies that will ultimately bring disease-modifying therapies to the market. And that support from the physician or from the patients has been echoed by many physicians who are interested to start working with the test. We understand there's still work to do. So, for us, we were able to demonstrate about a 0.88 ROC concordance with amyloid, a very high concordance. And interestingly, the amyloid measurements by PET are not perfect. So we think that the performance of the test is actually quite good. And we anticipate that we'll ultimately get the FDA approval. We have a breakthrough device designation. We're working towards that. And we understand that some physicians may want to see the FDA approvability, but at this point, we have the CLIA certification, very high quality standards, and very compelling clinical data that we felt it was really important to be able to start providing this tool. And we still feel like we're in the early days because we have a lot more to learn about this marker, how it's going to be used in clinical practice. But importantly, we think we have a really important way to now identify early amyloid pathology that will then aid in diagnosis for Alzheimer's disease and related neurodegenerative disorders. I guess my last comment is that we understand that this is likely to be a multimodal approach. There's going to be a need for not only plasma-based biomarkers, but some of the other biomarkers that have been discussed here, and the ability to measure additional analytes will help inform about really provide a compelling picture of what the patient's brain health looks like. So, we believe that we can detect amyloid pathology very early, but that's only one part of the puzzle. We're building out the mass spec platform, which we're very excited about, using the mass spectrometer to be able to measure these other analytes, neurofilament, phosphorylated tau forms, other tau forms that may be more closely integrated with disease progression as opposed to just being indicators of amyloid pathology, and helping in differential diagnosis. So we kind of see a future where people will be able to get their disease diagnosed with proper clinical assessments, these biomarkers, and then be able to use these markers in a way to track disease progression and review progress or responses to therapies.
Great, thanks very much for those very enlightening comments, and I'd really like to thank all of you for not only your terrific responses, but even more importantly for the outstanding work all of you are doing. I'd now like to segue into sort of a roundtable discussion. We've also received some questions from the audience, and some of those questions I'd like to start with would actually indirectly all start to touch on already. I think one of the things Gil was just mentioning that, and Mark also mentioned that basically patients want to know. Sometimes the medical profession can be a little bit paternalistic, but I think the evidence is overwhelming that people want to know. And, in addition to knowing, they can start to do something about it. Having said that, I do think there are some concerns about privacy and ethics, and concerns that most of us here would never even dream that this could happen, and yet we've got to be cognizant it could. People being denied medical insurance coverage because they may have a high risk score, etc. So I'd like us to start to just talk briefly about that, and maybe Richard, if you don't mind, I'll start with you. What are your thoughts about privacy and ethics concerns when it comes to some of this testing and how we as a field can try to ensure they don't get misused?
I think it's a very important point. The comment that's come from several participants in the group today is that patients and relatives really want to know about risk and what their future holds. So there's no holding this back. And many physicians that we talk to are confronted with individuals who've already obtained some of this data through direct-to-consumer type testing. So they might come armed with their ApoE status already and wanting to know how to interpret that and what it might mean for them. So there's a real thirst out there. And of course, although we're kind of in the front line because we're talking about genetics, once you accept that a disease had a strong genetic component, it really doesn't matter which modality you're working with, whether it's a blood biomarker, a CSF, or an imaging marker, you're still talking about a disease with a very strong genetic component. So I think those confidentiality issues and perhaps the insurance or medical sort of healthcare coverage type issues are the same regardless of the technologies you use. But, we just have to find a way. And, there are all sorts of ways to deal with this. I think in Europe, where we have the GDPR regulations, that sets a really very strong framework for how to deal with this and of course, any other confidential information that we just have to fit within. But what we're finding, and as Joel was saying, we're talking about introducing our test this month actually as a CE mark product in Europe and through a clear LDT partnership in the US. There's a real appetite there. Clinicians want to be able to provide this to their patients so that they can have that conversation and have the conversation about risk, but then the actionability. So you're in a high risk group, you don't yet have, advanced symptoms. Here's some steps we can take. So I think it's all about having an actionable outcome to whatever platform you're working with.
Yeah, very thoughtful response. And I think, as I said, it behooves us all to make sure while we're advancing these technologies, we have some of these considerations front and center so we can make sure our patients and society benefit. Yeah, Richard, you mentioned you use a term LTD. Just wondering if you could expand on that.
LDT, a laboratory developed test. So it's under the clear regulations. It's very much as Joel was describing. It's really about the technical and I think Rianne as well, the technical validation components to make sure that the same sample, it could be tested over and over again under slightly different conditions and you still get a robust answer at the end of it. So it's all around being able to trust the output of the test at the end of that validation period. And that set of experimental data is submitted to the appropriate regulatory sort of authority or overseer, and then that's used as part of the sort of quality management of that test delivery. And I think it's really important as well to note, we've been in discussion with the FDA, and the FDA have said, well, look, don't forget about the clear LDT route. This is a relatively quicker route to get to the market before you go down the full de novo 510K or whatever FDA route may be most appropriate for your product, and there's a need for it out there. So they were actually encouraging us to think about this rather than putting up any barriers. So this idea that you've got to keep everything quiet from the FDA and so you treat them as a kind of them and us dynamic, that really isn't true in our experience. They've been very, very helpful and very willing to guide.
Yeah, no, I agree completely. I think regulators really are a key part of the equation, and from my experience as well, they've been very thoughtful and very aligned. There's a million questions we could ask, but I'll focus on a few more, and I'll ask you to keep your responses brief so we can get as many of them as possible. Many of you touched on how combination approaches may fit into the ecosystem, so maybe we could expand a little bit on that. Rianne, maybe I could start with you. How do you see some of the work your company is developing fitting into the ecosystem with other approaches?
Sure. I think that there's just so many options for these combination approaches. One of them I mentioned was plasma being a funnel for CSF and PET testing, but also genetic testing. And being higher risk, and then starting to develop those memory problems, and then that leading you to want further diagnostic testing is critical. And in addition to that, not just having the initial diagnostic test, but also having those progression markers like p-tau, where you can really watch those disease-modifying therapies and see if they're actually helping the patient or if they should switch to another drug. Of course, those are all future hopes and dreams, but...
Great, thank you. And Steve, the same question, if you don't mind. How do you see combination approaches?
I see it two ways. One is amongst multiple blood biomarkers, to the points that Rianne just made, with respect to how we combine those intelligently, making sure we don't overfit data, when we're trying to produce results. The other is in combination with retina. So we've done some testing to that effect, and we're picking up, it's still early days, but we're picking up maybe something on the order of 8 points in AUC when we put retina and blood together orthogonally. So that's encouraging if we want to ultimately get to a point, to Tobias's point, where we may want to, long in the future, replace PET, if that's possible, if we can get our performance well into the 90s by this combination approach. So this is one of the approaches. It doesn't have to be that way for what we're doing. It can be blood only, it can be retina only, or the combination. We're going to let the data, tell us and inform us on how that should be. But that's how we view it, and that's the test that we're currently undertaking in the various studies that we have.
Great. And Mark, your company is one of the pioneers in brain PET imaging. Also, I want to ask you the same question, how you see the potential linkage or synergies of some of these combination approaches with brain imaging?
Oh, I think you've correctly identified that that while the PET studies are often sort of the gold standard and have some of the most compelling evidence for comparison to pathology, the very, very practical nature of getting that out there and the cost and the greater expense means that the blood tests in particular should be able to offer so much in the way of a much, much broader funnel. And we've been talking about them, in the A-beta 4240 are just huge advance, and as Joel has talked about. But we haven't mentioned, the p-tau 217 with the recent paper in JAMA by the group, Oscar Hansen groups again, is also another big step towards looking at the other pathology and more of a potentially sort of a rule in. And I think that, but the reality of blood tests like this is that there will be the really, really obviously negative people, which we hope will mean very low chance of having pathology, and the really obviously high, which will have a very high chance. But we've got to, I think, be very transparent as companies and as a community to what the data is for, what the real data is for being in between and being uncertain. And so that doctors and patients and families can say, well, if it's a lifestyle change, maybe I don't need any more change. This is enough to provoke me. But if it's going on a medication or if it's going on for additional testing, it's very important to have that data readily available, not just sort of like a test. What I'm realizing now is simply saying sensitivity, specificity is not enough. There's going to be gray zones on these types of tests, and we need to talk about what happens to people in those gray zones. And if we define them and are transparent as we get this data, and Lilly is very interested in understanding this, not only for the sake of potential having a disease modifier, which we're really, really hopeful for, but for the sake that even understanding how to get people into clinical trials means that you have to measure these things. So as people develop data sets and get LDTs on the market, the more that we can actually see the actual raw data and understand how many people were in the intermediate zone, what happened to them, I think that'll be key to building this combination, this funnel, really having a scientifically data-led funnel for our both clinical use and clinical trial use.
Great, thank you. I'd like now to turn to a question we received from the audience, and perhaps I could initially target it to Tobias. So the question basically is that, following the publication from Drs. Tansey and Moore demonstrating or suggesting the antimicrobial activity of A-beta, that question is anyone considering examining or developing infection biomarkers in parallel to some of the other biomarkers we're talking about? And Tobias, as I said, maybe I could start with you.
Yeah, that's a very interesting question. I'm not sure if I'm the best person to answer that, but I've been following this for quite some time, and I'm quite interested and intrigued by it because it provides a couple of explanations for things that the AD field, I think, has been struggling with explaining in the last decades. We have not done any particular work on that, but I think one thing that we can do now that we embark more into measuring plasma is that we take already developed and marketed assays that might shed a light on bacterial infections and just plug them into the analysis that we're doing with the more Alzheimer or dementia-related biomarkers. And by that, just generate additional data and open the door to be surprised to maybe find something here. So that's, to me, the only angle that I can see how our work can actually help with that hypothesis.
Okay, great. Obviously it's a very interesting suggestion, and I'm just wondering if anyone else, if you've got a brief response, if you're already doing some work related to infection biomarkers in addition to what we might call the more traditional ones. Okay, I'd like to now move on to another question. As we said earlier, obviously COVID-19 has been devastating, but are there any lessons from COVID-19 that we can sort of take, for example, opportunities to accelerate clinical trials or cut out bureaucracy that can bring to the dementia field? You heard earlier from Arnaud about, the sort of staggering, development of vaccines within 1 year that we would have thought is unfeasible, and yet it's happening. And so maybe I'll start with Mark, if there are any insights you have.
Yeah, it has made us think long and hard. I think one of the things that's been alluded to is the more we can be creative about doing decentralized trials where we don't require back and forth to a CMO, some sort of CRO site for the investigator site for the patient, the more we can do in their house. Blood tests would just be huge in being able to screen for patients, and they don't even have to leave their home. If there was a nurse, these remote ways of doing cognitive assessments and functional assessments would be huge. So I think that's one of the two things that we're taking away is really important, is to develop that. That doesn't mean that you go back to the old way once the pandemic is over, and we all hope for that to happen very soon. It means that actually you might be able to recruit patients much, much easier and be able to crack this incredible problem of getting patients into trials right now of something that is so important, but yet takes so long. There are many times that trials you have to be on drug for 18 months or 2 years. Sometimes it takes 18 months to 2 years just to recruit the patients for the trial. And that's the unnecessary part. And then the second thing I think we've learned, and a little bit of the same message I said before, but it's pertinent because of COVID, is look at the amount of worry people have about rushing a vaccine for COVID, and how much the transparency it helps. In other words, as these companies realize that they need to be as transparent as possible, put as much of the data out there, the protocol out there, exactly what type of patients were enrolled, how they parse their data into subsets to calculate the numbers, that type of transparency, I think, will go a long way to help people accept the results of the vaccine, and I think will go a long way to accept the results of any diagnostic or therapeutic trial on Alzheimer's disease.
Great, well said. Another interesting question from the audience, and perhaps I'll address it to you, Steve, to begin with. With the advent of AI prediction through linguistics, financial modeling, these things are proving to be potentially early useful diagnostic markers. Some of you may know that thanks to Arnaud and George, there's also a Davos Alzheimer's collaborative underway, and as part of that, we're really taking these digital markers very seriously. Wondering if you have some thoughts about, potentially combining some of these digital markers with either the fluid-based or imaging biomarkers to really maximize predictability?
Yeah, I think they're very important, and I think they should be considered. And even to Tobias's point about where you already have a platform, like we do as well, where you can add other markers and really do these tests and understand what their contributions are. So whether it's natural language, cognition, genetic information, I think all of that. I think the thing we all have to be mindful of, which we sometimes see even in high impact journals, is overfitting of data. Too many variables, not large enough sample where you can get the result you want. And it's great for academic papers, but we end up fooling ourselves. So for those of us in companies that are producing real products, it's important that we make sure that we pass those false discovery tests and permutation tests and other things. So I think 100% it should be all of the above, but let's be careful when we analyze it to make sure when we include them that we're doing it intelligently. But it's an amazing emerging field, especially with respect to some foundational work originally done by IBM in just voice recognition and other aspects of deep learning and AI is very well suited to some of this in terms of early detection. Non-specific, but can add towards understanding how it may contribute to disease progression.
Yeah, I think those points are very well taken, and I'll just amplify one of the things you said. As I said previously, sometimes people are wondering why some of these things take long. The digital technology revolution is potentially going to really help us here. But as Steve said, let's make sure if we're going to be, potentially even making decisions about, patient healthcare or understanding who's at risk. Let's make sure we do it correctly. And I think that's one of the things we'd all agree to. I think we could go on for several hours given the wealth of information and expertise on this panel. But I think in the interest of time, but I'd like to give all of you, it's basically sort of a minute to wrap things up with, where do you think we are and what one action do you think we should take to produce the greatest result for, making a difference for humanity? And maybe I'll start with you, Joe.
Sure, thank you. Yeah, I come from the field of cardiovascular medicine, and one thing that the field has done very well in cardiology is really map the longitudinal progression of disease, understand risk factors, and develop markers that are highly indicative of likely future events. And I think that we're now in the field of neurology and Alzheimer's disease specifically, we're now embarking on a time where we're going to be able to predict and identify the earliest beginnings of pathology 10, 15, 20 years before the onset of symptoms. That is absolutely profound. And if we can draw some parallels to what happened in cardiovascular medicine, when that happened, we were able to reduce long-term cardiac events by 50% because we were able to intervene earlier. And I think even the FDA has kind of endorsed this concept that the earlier you can identify pathology before irreversible neurodegeneration occurs, the more likely it is that you're going to have a therapeutic benefit. I think the other key point is, so the importance of continuing to try to push the science and demonstrate the value with well-conducted clinical studies to predict downstream events will be really important to the field. And we're now embarking in that time with these biomarkers from multiple different platforms. I think the final point is education. There is a lot of misunderstanding or general biases by both the clinical community and even among patients who have these preformed ideas as to what it means to obtain a diagnosis of Alzheimer's disease. And there's no refuting the fact that it's a horrific disease, but the ability to, again, become more knowledgeable and to understand that there are things you can do. And we're starting to get that data to show that by being more proactive, you can have a benefit to your family and to your own personal health by being more knowledgeable.
Thank you. Very well said. Tobias, same thing, final comments and a call to action?
Yeah, I think it's very exciting times in Alzheimer's disease and also specifically in biomarkers. I think our main goal is to transform the diagnostic pathway to make it less complicated, more cost effective. Blood biomarkers play an important role here. Also empowering the GP to actually front load some of the activities that are currently being done in clinics, which seem to be the bottleneck in getting an Alzheimer diagnostic to the people at the right time in their disease. So I think this whole field will move very quickly. But I still want to caution that and manage expectations, because I do think that it will take another 5 to 10 years before we have really high quality assays that can be used in clinical routine on a very large scale globally, and then also have those adopted in guidelines and be appreciated by the field. So I think that's not something we can flip a switch on and have in a year or two. I really do think we need to think in 5 to 10 years. timelines on that, unfortunately.
We're short on time, so I'll continue quickly if you don't mind, everyone. Rianne, your call to action.
Sure, and I agree with Tobias and Joel. Just having cost-effective, robust, accurate, high-performing diagnostics for patients. But also the most important thing is educating these practitioners and neurologists about how the diagnostics and biomarkers can provide value to their patients and how to use the biomarkers and PET tracers properly.
Great. Those are all fantastic comments, and I'd like to echo everything you said. I'd like to thank all our terrific panelists. I think, as you can see, there's reasons for us to be, as a society, confident about making